The ribosome rotates with mechanical precision, its two subunits sliding past each other like gears in a molecular machine. Transfer RNA molecules dock and undock in perfect sequence, each carrying its designated amino acid to the growing protein chain. The entire assembly operates at speeds that would make any human-engineered factory seem clumsy by comparison, translating genetic instructions into functional proteins at a rate of 15 amino acids per second, with an error rate of less than 1 in 10,000. This is not the product of billions of years of gradual evolution. This is what life looked like on Earth 4 billion years ago, when our planet was barely 500 million years old. A cosmic eye blink after the hellish bombardment that had sterilized the surface and boiled away the early oceans. Somehow, in this impossibly brief window of geological time, matter had organized itself into systems of such staggering complexity that they could reproduce, evolve, and ultimately give rise to every living thing on our planet. The speed of this transformation defies everything we know about probability, chemistry, and time. It's as if someone had scattered the components of a Boeing 747 across a field and returned the next morning to find it fully assembled and ready for flight. Yet this is precisely what conventional theories of abiogenesis ask us to believe, that life spontaneously emerged from non-living matter through purely random chemical processes operating in the primordial soup of early Earth. The mathematical implausibility of this scenario has haunted biologists since they first began to understand the true complexity of even the simplest living cells. A single functional protein containing just 150 amino acids arranged in the correct sequence represents odds of roughly 10 to the 164th power against its random formation. To put this in perspective, the entire observable universe contains approximately 10 to the 82nd power atoms. The probability of assembling even one functional protein by chance is so vanishingly small that it makes winning the lottery every day for 1 billion years seem like a virtual certainty. Yet life on Earth requires not just one functional protein, but thousands of them, all working together in precisely coordinated systems. The simplest known bacterium contains over 400 genes, each encoding complex molecular machines that perform specific functions essential for survival. These systems exhibit what biochemists call irreducible complexity. Remove any component and the entire system fails. There are no intermediate stages, no simpler versions that could serve as evolutionary stepping stones. This presents what scientists call the genesis problem. How did such complex integrated systems arise so quickly in Earth's early history? The traditional answer, gradual evolution from simpler precursors, runs headlong into the mathematical wall of probabilistic impossibility. Even with billions of years available, the odds against the spontaneous formation of life remain so astronomically large that many researchers have begun looking elsewhere for solutions. The search for answers has led to one of the most provocative hypotheses in modern science: panspermia. The term, coined by Swedish chemist Svante Arrhenius in 1903, literally means seeds everywhere. According to this theory, life didn't originate on Earth at all, but arrived here from somewhere else in the cosmos, either through natural processes involving meteorites and comets, or through the deliberate actions of an advanced extraterrestrial civilization. The implications are staggering. If panspermia is correct, then every living thing on Earth, every tree, every animal, every human being, carries within its cells the molecular legacy of another world. We are not the products of terrestrial chemistry, but descendants of cosmic travelers who made the ultimate journey across the vast emptiness of interstellar space. Our DNA contains not just the history of evolution on Earth, but potentially the genetic memories of civilizations that flourished among distant stars billions of years ago. This possibility first gained scientific credibility through the work of Francis Crick, the Nobel Prize-winning co-discoverer of DNA's double helix structure. In 1973, working with biochemist Leslie Orgel, Crick published a paper in the journal Icarus that proposed what they called directed panspermia, the hypothesis that life on Earth was deliberately planted here by an advanced alien civilization. Crick's reputation in molecular biology gave the idea immediate attention, but it was his reasoning that made it compelling. As someone who had spent decades studying the molecular machinery of life, Crick understood better than most just how improbable its spontaneous emergence really was. The genetic code itself, the system by which DNA sequences are translated into proteins, showed signs of optimization that suggested design rather than chance. It was almost as if someone had carefully engineered the fundamental language of life to be as efficient and error-resistant as possible. The timing of life's appearance on Earth provided another piece of the puzzle. Geological evidence suggested that complex cells existed within the first billion years of our planet's formation, and possibly much earlier. This left little time for the gradual chemical evolution that conventional theories required. If life had originated elsewhere and travelled to Earth as fully formed organisms, however, the timing problem disappeared. The billions of years needed for life's initial emergence could have occurred on some other world, while Earth served merely as a new habitat for already evolved creatures. The discovery of extremophile organisms, life forms that thrive in conditions previously thought impossible for biology, further strengthened the panspermia hypothesis. These remarkable creatures found in boiling hot springs, highly acidic environments, and even in the vacuum of space, demonstrated that life was far more resilient than previously imagined. If organisms could survive such extreme conditions on Earth, perhaps they could also survive the harsh environment of interstellar space during journeys lasting millions of years. Recent discoveries have made the possibility even more plausible. Meteorites recovered from Antarctica contain complex organic compounds that formed in space, including amino acids, the building blocks of proteins. Some of these amino acids show what scientists call a left-handed preference, identical to that found in Earth's biology, suggesting a possible connection between terrestrial life and its cosmic precursors. The Murchison meteorite, which fell in Australia in 1969, contained over 70 different amino acids, including many not found in Earth's biosphere. More remarkably, these compounds showed isotopic ratios that proved their extraterrestrial origin. The meteorite had carried organic chemistry from space directly to Earth's surface, demonstrating that the basic ingredients of life could indeed survive interplanetary travel. Space-based telescopes have detected complex organic molecules in interstellar gas clouds, stellar nurseries, and even in the atmospheres of distant planets. The Spitzer Space Telescope identified polycyclic aromatic hydrocarbons, complex carbon compounds, throughout the galaxy. These molecules form the backbone of many biological systems, and their widespread presence suggests that organic chemistry is a universal phenomenon rather than a peculiarity of Earth. But perhaps the most compelling evidence for panspermia comes from the study of life's emergence patterns. Whenever life gains access to a new environment on Earth, whether through continental drift, climate change, or human activity, it spreads and diversifies with remarkable speed. This suggests that life is fundamentally opportunistic, ready to exploit any available niche as quickly as possible. If this pattern held true on a cosmic scale, advanced life forms would naturally seek to colonize every suitable planet they encountered. The question becomes, what kind of civilization would have the motivation and capability to seed life throughout the galaxy? The answer may lie in understanding the ultimate fate that awaits every technological species. Advanced civilizations face inevitable extinction from various sources: their host star's evolution, cosmic catastrophes, or even the heat death of the universe itself. For a dying civilization with millions or billions of years of accumulated knowledge, the prospect of total annihilation would be unthinkable. Such a civilization might view the seeding of younger worlds not as conquest, but as a form of cosmic reproduction, a way to ensure that intelligence and consciousness would continue to emerge throughout the galaxy long after their own world had turned to dust. They would be motivated not by colonialism, but by the deepest biological imperative, the drive to perpetuate life itself. The technical challenges would be enormous, but not insurmountable for a sufficiently advanced species. Spacecraft capable of interstellar travel would need to maintain biological samples in viable condition for millions of years. But recent advances in synthetic biology suggest that entire ecosystems could potentially be encoded in synthetic DNA and reconstructed upon arrival at target worlds. Instead of transporting actual organisms, alien seeders might have sent sophisticated genetic programs capable of bootstrapping life from local materials. This possibility raises profound questions about human purpose and destiny. If we are indeed the products of alien seeding, what does that mean for our understanding of evolution, consciousness, and our place in the cosmic order? Are we simply accidents of chemistry, or were we specifically designed by predecessors who recognized something precious in the phenomenon of self-aware matter. The search for answers has become one of the most important quests in modern science. Astrobiologists scan the skies for signs of extraterrestrial life, while molecular biologists dissect genomes looking for signatures that might reveal non-terrestrial origins. Each discovery, from organic compounds in meteorites to extremophile organisms in Earth's most hostile environments, adds another piece to a puzzle that could fundamentally reshape our understanding of life's place in the universe. Yet the Genesis problem remains unsolved. Despite decades of research, no one has successfully demonstrated how complex life could arise spontaneously from non-living matter under realistic conditions. Laboratory experiments designed to simulate early Earth's environment have produced only simple organic compounds, nothing approaching the integrated complexity of even the most primitive living cells. The mathematical obstacles have not disappeared with better understanding of chemistry and molecular biology. If anything, the discovery of life's true complexity has made spontaneous emergence seem even more improbable than early researchers imagined. Every cell contains thousands of interconnected molecular machines, all of which must function together perfectly for the organism to survive. The odds against such systems arising by chance remain as astronomical as ever. This mathematical crisis in biology has created an opening for alternatives that were once considered too speculative for serious scientific consideration. Panspermia, whether natural or directed, offers a potential solution to the probability problem by moving life's origins to somewhere with more time and resources available. If life originated in an environment with billions of years and countless planets to work with, the mathematical obstacles that seem insurmountable on early Earth become merely challenging on a galactic scale. The implications extend far beyond biology into the realms of philosophy, theology, and human meaning. If life on Earth was seeded by alien intelligence, then our existence represents the fulfillment of another civilization's deepest hopes and dreams. We're not cosmic accidents, but intended consequences, the children of minds that gazed at the stars billions of years ago and decided that intelligence should not perish from the universe. But to understand whether life came from the stars, we must first confront how impossible life's emergence actually is. The spark jumped between the electrodes with a satisfying crack, sending voltage surging through a mixture of methane, ammonia, and hydrogen gas. In his cramped laboratory at the University of Chicago in 1953, graduate student Stanley Miller watched as his apparatus, a sealed glass system designed to simulate Earth's primordial atmosphere, began to transform clear gases into a brownish soup of organic compounds. Within days, the experiment had produced amino acids, the building blocks of proteins. Life, it seemed, could indeed emerge from non-living matter given the right conditions and sufficient time. The Miller-Urey experiment, conducted under the guidance of Nobel laureate Harold Urey, electrified the scientific community and launched the modern field of origin of life research. Here was proof that complex organic molecules could form spontaneously from simple gases under conditions thought to resemble early Earth. The experiment appeared to vindicate Charles Darwin's speculation about life arising in some warm little pond through purely natural processes. News of the breakthrough made international headlines. Time magazine proclaimed it evidence that scientists were close to creating life itself in the laboratory. The optimism was infectious. If amino acids could form so readily under simulated primitive conditions, surely the emergence of living cells was just a matter of scaling up the process and adding more time. But as researchers attempted to build on Miller's success, troubling problems emerged. The amino acids produced in the experiment were a 50-50 mixture of left-handed and right-handed forms, while all life on Earth uses exclusively left-handed amino acids. The conditions required to produce organic compounds, a highly reducing atmosphere with no oxygen, appeared inconsistent with geological evidence about early Earth's actual environment. Most critically, the experiment had produced only the raw materials of life, not life itself. The gulf between amino acids floating in solution and functioning living cells remained as vast as ever. Fred Hoyle, the brilliant British astronomer who had coined the term Big Bang as a dismissive nickname for cosmic expansion theories, turned his attention to biological origins in the 1970s with characteristic iconoclasm. Working with mathematician Chandra Wickramasinghe, Hoyle calculated the probability of even the simplest living cell arising through random chemical processes, The numbers were staggering. A typical bacterial cell contains about 2,000 different enzymes, each consisting of roughly 300 amino acids arranged in a specific sequence. The probability of getting just one functional enzyme through random assembly was approximately 1 in 10 to the 130 power. For all 2,000 enzymes to form simultaneously, the minimum required for cellular function, the odds became 1 in 10 to the 260,000 power. To put this impossibility in perspective, Hoyle famously compared it to the probability of a tornado sweeping through a junkyard and accidentally assembling a Boeing 747. The comparison wasn't hyperbole. If anything, it understated the mathematical challenges facing spontaneous biogenesis. A jumbo jet contains roughly 6 million parts, while a bacterial cell contains billions of precisely arranged atoms functioning as integrated molecular machines. The protein problem was just the beginning. Even if functional proteins could somehow arise by chance, they would need to be protected by cellular membranes, supplied with energy through metabolic pathways, and equipped with genetic systems capable of reproduction and evolution. Each of these requirements introduced additional layers of irreducible complexity that made spontaneous emergence even more improbable. The genetic code itself presented what some researchers called the ultimate chicken and egg problem. DNA contains the instructions for making proteins, but proteins are required to read and copy DNA. Ribosomes, the molecular machines that translate genetic information into functional proteins, are themselves made of proteins and RNA. The system appeared to require all its components to exist simultaneously, with no obvious pathway for gradual assembly. Walter Gilbert at Harvard University proposed a potential solution in 1986 with his RNA world hypothesis. Perhaps life had begun with RNA molecules that could both store genetic information like DNA and catalyze chemical reactions like proteins. These self-replicating ribozymes could have bootstrapped the first primitive life forms, eventually giving rise to the more sophisticated DNA protein systems we observe today. The idea gained support when researchers discovered naturally occurring ribozymes in living cells and successfully created artificial RNA molecules with catalytic properties in the laboratory. But the RNA world hypothesis faced its own mathematical obstacles. Gerald Joyce at the Scripps Research Institute calculated that a self-replicating RNA molecule would need to be at least 100 nucleotides long to function reliably. The probability of such a sequence arising by chance was approximately 1 in 10 to the 60th power, still astronomically unlikely, though less so than spontaneous protein formation. More troubling was the instability of RNA molecules under realistic prebiotic conditions. RNA degrades rapidly in water, especially at the elevated temperatures thought to characterize early Earth. Laboratory experiments showed that RNA chains longer than a few dozen nucleotides fell apart faster than they could be synthesized under plausible primitive conditions. The RNA world, if it had ever existed, would have been a fragile and short-lived phenomenon. As the challenges facing terrestrial abiogenesis mounted, researchers began looking more seriously at the timeline constraints imposed by Earth's geological history. The planet formed approximately 4.6 billion years ago, but remained uninhabitable for several hundred million years due to bombardment by asteroids and comets during the late heavy bombardment period. The earliest evidence of life, microscopic fossils in ancient rock formations, dated to approximately 3.8 billion years ago, just 700 million years after Earth became habitable. 700 million years might seem like ample time for life to emerge, but the number became less impressive when viewed against the mathematical obstacles involved. Even with the entire age of the universe, nearly 14 billion years, random processes would be unlikely to produce a single functional protein, let alone the integrated systems required for cellular life. The rapid appearance of complex life on early Earth began to look less like evidence for easy biogenesis, and more like evidence for a process that had already occurred elsewhere. The problem deepened when researchers discovered that even the simplest life forms were staggeringly complex. Mycoplasma genitalium, often cited as the organism with the smallest known genome, still contained 470 genes encoding essential cellular functions. Craig Venter's team at the J. Craig Venter Institute spent years systematically removing genes from this minimal organism to determine which were truly essential for survival. Their synthetic biology experiments revealed that even the most stripped-down artificial cell, required at least 370 genes to maintain basic life processes. Each of these genes encoded proteins containing hundreds of amino acids arranged in precisely defined sequences. The mathematical improbability of their simultaneous emergence on early Earth defied comprehension. It was as if matter had organized itself into the most complex structures in the known universe during Earth's cosmic infancy, accomplishing in a few 100 million years what random processes shouldn't be able to achieve in trillions of years. This temporal paradox extended beyond individual proteins to entire biochemical pathways. Photosynthesis, the process that powers most life on Earth, requires the coordinated action of dozens of enzymes working in precisely timed sequence. The photosystem complexes that capture sunlight and convert it to chemical energy are among the most sophisticated molecular machines ever discovered, yet geological evidence suggested they had evolved within the first 2 billion years of Earth's history. The emergence of oxygenic photosynthesis represented an even greater puzzle. Cyanobacteria, the first organisms capable of splitting water molecules to release oxygen, fundamentally transformed Earth's atmosphere and enabled the evolution of complex multicellular life. But the biochemical machinery required for water-splitting photosynthesis was extraordinarily complex, involving multiple protein complexes, cofactors, and electron transport chains. The system showed no obvious evolutionary intermediates. It appeared in the geological record as a fully functional innovation. Metabolic pathways presented similar challenges. The citric acid cycle, which extracts energy from nutrients in nearly all living cells, involves 8 precisely coordinated enzymatic steps. Each enzyme in the cycle is essential. Remove any one and the entire system fails. Yet the cycle appeared to be present in the earliest life forms, suggesting it had evolved during the initial emergence of cellular life rather than through later evolutionary refinement. The universality of certain biochemical features across all domains of life, bacteria, archaea, and eukaryotes, suggested these systems had been present in the last universal common ancestor, or LUCA, that gave rise to all modern organisms, but LUCA itself must have been an extraordinarily sophisticated organism, possessing complex genetic machinery, metabolic pathways, and cellular structures. The idea that such an organism could have arisen spontaneously from non-living matter seemed to push the boundaries of scientific plausibility. Even the basic chemistry of life presented obstacles that laboratory experiments couldn't overcome. The formation of peptide bonds between amino acids, the chemical links that hold proteins together, requires the removal of water molecules. But early Earth was covered by oceans, making water removal thermodynamically unfavourable. Researchers proposed various solutions involving mineral surfaces, temperature gradients, or chemical catalysts, but none proved capable of producing the long, complex proteins essential for cellular function. The chirality problem remained equally stubborn. Life uses exclusively left-handed amino acids and right-handed sugars, but chemical reactions under abiotic conditions produce equal mixtures of both forms. Some mechanism must have selected for one-handedness over the other, but no convincing explanation had emerged from decades of research. The homochirality of biological molecules appeared to be another feature that required explanation beyond simple chemistry. Lipid membranes, which enclose all living cells and define the boundary between life and environment, presented their own formation challenges. Cell membranes are not simple barriers, but sophisticated structures that selectively regulate the passage of molecules in and out of cells. They contain hundreds of different lipid types arranged in precise geometric patterns, often incorporating protein complexes that perform specific transport and signaling functions. Laboratory attempts to create self-assembling membrane systems had achieved only simple vesicles, microscopic bubbles that formed spontaneously under certain conditions, but lacked the complexity and selectivity of biological membranes. The gap between laboratory vesicles and functional cell membranes remained enormous, with no clear pathway for bridging the difference through purely chemical processes. The information problem posed perhaps the greatest challenge of all. Living cells store and process vast amounts of information in their DNA sequences, but information requires a code for its interpretation and machinery for its implementation. The genetic code that translates DNA sequences into proteins is itself a sophisticated information processing system, but its origin remained completely mysterious. information theorist Hubert Yockey calculated that the probability of the genetic code arising by chance was effectively zero. The code showed clear signs of optimization. It minimized the effects of copying errors and maximized the efficiency of information storage. Such optimization suggested selection pressure acting over long periods, but selection required reproduction, which required the very genetic machinery whose origin was in question. These mounting difficulties led some researchers to embrace increasingly exotic scenarios for life's emergence. The iron-sulfur world hypothesis proposed that life began at deep-sea hydrothermal vents, where metal sulfides could have catalyzed organic chemistry under high-temperature, high-pressure conditions. The metabolism-first hypothesis suggested that organized chemical reactions preceded the evolution of genetic systems, with metabolic networks somehow bootstrapping themselves into existence before developing the capacity for reproduction. But each alternative faced its own version of the probability problem. Complex chemical networks were no more likely to arise spontaneously than complex genetic systems. Moving the origin of life to exotic environments simply relocated the mathematical obstacles without resolving them. As the 21st century progressed, the gulf between laboratory demonstrations and realistic biogenesis scenarios continued to widen. Researchers could produce amino acids, nucleotides, and simple organic polymers under carefully controlled conditions, but they couldn't demonstrate the emergence of integrated living systems. The more scientists learned about cellular complexity, the more improbable spontaneous biogenesis appeared. The timing constraints became even more severe with improved understanding of early Earth conditions. The planet's surface had been repeatedly sterilized by massive asteroid impacts until approximately 3.8 billion years ago. Life must have emerged in the relatively brief window between the end of heavy bombardment and the first appearance of microfossils, a period that might have lasted only a few 100 million years. This temporal compression made the mathematical problems even more acute. Not only did complex life have to arise against astronomical odds, it had to do so very quickly on a geological timescale. The more researchers learned about both the complexity of life and the constraints of early Earth, the more mysterious life's rapid emergence became. The accumulating evidence pointed toward a conclusion that few scientists wanted to embrace. The spontaneous emergence of life from non-living matter, while theoretically possible, was so improbable that it might never have occurred anywhere in the observable universe. If life existed on Earth, and obviously it did, then perhaps it had originated elsewhere, under conditions more favourable than those that prevailed on early Earth. This realisation opened the door to possibilities that had once seemed like pure science fiction. If life was too complex to arise spontaneously, perhaps it hadn't arisen spontaneously at all. The meteorite hit the frozen ground of Antarctica with a sound like thunder, its dark mass contrasting starkly against the pristine white ice. For thousands of years it lay buried beneath accumulating snow, preserving within its rocky matrix a cargo more precious than any earthly treasure. Organic compounds that had formed in the depths of interstellar space billions of years before Earth itself existed. When researchers from the Antarctic Search for Meteorites program finally recovered the specimen in 1969, they had no idea they were handling a time capsule from the dawn of the solar system. The Murchison meteorite, as it came to be known, would revolutionize our understanding of cosmic chemistry and provide the first concrete evidence that the building blocks of life could survive journeys across the void of space. In sterile laboratories around the world, scientists began the painstaking process of analysing the meteorite's composition. What they found defied every expectation about the sterility of space. The Murchison meteorite contained over 70 different amino acids, far more varieties than the 20 types used by terrestrial life. More remarkably, these compounds showed isotopic signatures that proved beyond doubt their extraterrestrial origin. The meteorite had carried organic chemistry from space directly to Earth's surface. The discovery shattered the assumption that complex organic molecules required biological processes for their formation. Here was proof that the fundamental chemistry of life could arise through purely abiotic processes operating in the harsh environment of space. But even more intriguing were the subtle patterns hidden within the meteorite's organic inventory. Analyses by researchers at NASA's Goddard Space Flight Center revealed that the amino acids in the Murchison meteorite showed a slight preference for left-handed forms, the same chirality bias found in all terrestrial life. The effect was small, only about 15% excess of left-handed molecules, but it was statistically significant and consistent across multiple independent measurements. Somehow, processes operating in space had produced the same molecular handedness that characterized biology on Earth. This finding suggested either an extraordinary coincidence or a fundamental connection between cosmic chemistry and terrestrial life. If extraterrestrial processes naturally produced left-handed amino acids, perhaps Earth's biology had inherited its chirality from cosmic precursors rather than developing it independently through unknown selection mechanisms. The implications deepened as researchers analysed other meteorites recovered from around the globe. The Tagish Lake meteorite, which fell in Canada in 2000, contained complex organic compounds including nucleotide bases, the fundamental components of DNA and RNA. The Murray meteorite showed evidence of polycyclic aromatic hydrocarbons, complex carbon structures that form the backbone of many biological molecules. Each new analysis reinforced the same startling conclusion. Space was not the sterile void that scientists had imagined, but a vast chemical laboratory where organic synthesis proceeded through mechanisms that remained largely mysterious. The molecular building blocks of life were not unique to Earth, but appeared to be common throughout the cosmos. Laboratory simulations of interstellar conditions began to reveal how such complex chemistry might arise in space. Ultraviolet radiation from stars could drive chemical reactions in ice-coated dust grains floating in molecular clouds. Cosmic rays provided energy for bond formation between simple molecules. The extreme cold of space, rather than inhibiting chemistry, actually promoted the formation of complex organic compounds by preventing their thermal breakdown. Max Bernstein at NASA Ames Research Center conducted groundbreaking experiments that recreated the conditions inside interstellar ice grains. By exposing mixtures of water, methanol and ammonia to ultraviolet radiation at temperatures near absolute zero, his team produced dozens of organic compounds including amino acids, sugars, and nucleotide bases. The experiments demonstrated that the chemistry of life could indeed occur in the apparent emptiness of interstellar space. But the discovery of organic compounds in meteorites was just the beginning. Space-based telescopes began detecting complex molecules in regions previously thought too hostile for any chemistry at all. The Spitzer Space Telescope identified polycyclic aromatic hydrocarbons throughout the galaxy, from stellar nurseries to the outer reaches of spiral arms. These molecules, containing dozens of carbon atoms arranged in intricate ring structures, represented some of the most complex chemistry ever observed in space. Radio telescopes detected even more sophisticated molecules in interstellar gas clouds. The Atacama Large Millimeter Array identified glycolaldehyde, a simple sugar, in a star-forming region 400 light-years from Earth. Other observations revealed formamide, a molecule considered a potential precursor to nucleotide bases, in multiple locations throughout the galaxy. The inventory of space-based organic chemistry grew longer each year. Methanol, ethanol, acetaldehyde, formic acid, hydrogen cyanide, and hundreds of other compounds that played essential roles in terrestrial biochemistry. The universe appeared to be conducting the same types of chemical reactions that occurred in living cells, but on scales spanning light-years and timescales lasting millions of years. Even more remarkable were discoveries within our own solar system. The Cassini mission to Saturn revealed that the Moon, Enceladus, possessed a subsurface ocean containing complex organic compounds. Geysers erupting from the Moon's south pole carried these molecules into space, where they could be analysed by the spacecraft's instruments. The organic inventory included compounds with carbon-nitrogen bonds, suggesting chemistry that could potentially support biological processes. Titan, Saturn's largest moon, proved to be a world where organic chemistry operated on a planetary scale. The thick atmosphere contained methane, ethane, and complex hydrocarbons that formed clouds, rain, and even lakes on the surface. The Huygens probe, which landed on Titan in 2005, detected dozens of organic compounds in the atmosphere and on the surface, including some that served as precursors to amino acids under appropriate conditions. Jupiter's moon, Europa, showed signs of organic chemistry occurring at the interface between its icy surface and subsurface ocean. Spectroscopic observations revealed sulfur compounds that could result from chemical reactions between organic molecules and the Moon's radiation-processed ice. The chemistry appeared complex enough to potentially support metabolic processes if life existed in Europa's hidden ocean. But perhaps the most intriguing discoveries came from studying extremophile organisms on Earth, life forms that thrived in conditions previously thought impossible for biology. These remarkable creatures demonstrated that life was far more resilient and adaptable than anyone had imagined, capable of surviving in environments that closely resembled conditions found throughout the solar system. Deinococcus radiodurans, discovered in canned meat that had been sterilized with gamma radiation, could withstand radiation levels thousands of times higher than would kill most organisms. The bacterium possessed extraordinary DNA repair mechanisms that could reconstruct its genome even after it had been shattered into hundreds of fragments by radiation damage. Such capabilities suggested that life might survive exposure to the cosmic rays that pervade interstellar space. Tardigrades, microscopic animals also known as water bears, proved even more resilient. These tiny creatures could survive complete dehydration for decades, temperatures approaching absolute zero, and even the vacuum of space. European Space Agency experiments demonstrated that tardigrades could survive and reproduce after being exposed to the harsh conditions of low Earth orbit for 10 days. The discovery of life in Earth's most extreme environments revealed that biology could potentially operate in conditions found throughout the solar system and beyond. Hyperthermophile bacteria thrived in boiling water around deep-sea hydrothermal vents, suggesting that life might exist in the subsurface oceans of icy moons. Acidophile organisms flourished in environments with pH levels that would dissolve metal, indicating that life might survive in the acid clouds of Venus or other chemically hostile worlds. Psychrophile bacteria grew and reproduced at temperatures well below freezing, remaining active even when encased in ice for thousands of years. Researchers discovered living bacteria in permafrost samples that had been frozen for over 500,000 years, demonstrating that life could potentially survive the long journey between star systems if protected within ice or rock. These discoveries transformed scientific thinking about the possibility of panspermia, the transfer of life between worlds through natural processes. If organisms could survive such extreme conditions on Earth, perhaps they could also survive ejection from their home planet, journey through interstellar space, and landing on new worlds. The mechanism for such transfers had been identified through studies of Martian meteorites found on Earth. Powerful impacts could blast rocks from planetary surfaces with enough velocity to escape gravitational pull and travel between worlds. Computer simulations showed that material ejected from Mars could reach Earth within a few million years, while some debris might take hundreds of millions of years to complete the journey. Analysis of Martian meteorites revealed that some contained organic compounds that had formed on Mars before the rocks were blasted into space. While these compounds were not definitively biological in origin, they demonstrated that complex organic chemistry could survive the violent process of impact ejection, interplanetary travel, and atmospheric entry. The discovery of organic compounds in comets provided another potential mechanism for delivering life's building blocks to planetary surfaces. The Stardust mission returned samples from Comet Wild 2 that contained glycine, the simplest amino acid used by terrestrial life. The European Space Agency's Rosetta mission detected 16 organic compounds in the atmosphere and on the surface of Comet 67P/Churyumov-Gerasimenko, including 4 compounds that had never been detected in comets before. These findings suggested that comets might serve as delivery vehicles for organic chemistry throughout the solar system. During the late heavy bombardment period, billions of comets and asteroids struck the inner planets, potentially delivering vast quantities of organic compounds to their surfaces. If some of these compounds were sufficiently complex to kickstart biological processes, comets might have seeded multiple worlds with the ingredients for life. But the most provocative evidence for cosmic connections to terrestrial biology came from studies of the genetic code itself. The universal genetic code used by all life on Earth showed patterns that seemed almost too perfect to have arisen through random evolutionary processes. Theoretical calculations by researchers at Los Alamos National Laboratory revealed that the genetic code was optimized to minimize the effects of copying errors and chemical damage. Of all possible ways to assign DNA triplets to amino acids, the actual genetic code ranked in the top 1% for error minimization. The probability of such optimization arising by chance was less than 1 in a million. Even more intriguing were mathematical patterns embedded within the genetic code that seemed to echo fundamental physical constants. Some researchers claimed to find relationships between codon assignments and the mass ratios of elementary particles, though these findings remained controversial and disputed by other scientists. The universality of the genetic code across all domains of life suggested it had been frozen very early in biological evolution, possibly during the emergence of the first living cells. But why had evolution settled on this particular code rather than one of the millions of alternatives? The optimization for error correction suggested some form of selection pressure, but selection required reproduction, which required the genetic machinery whose origin remained mysterious. These patterns led some researchers to speculate that the genetic code might preserve evidence of its extraterrestrial origins. If life had been designed by an advanced civilization, they might have embedded mathematical signatures within the genetic code that could be recognized by any species sophisticated enough to decode genomes. Carl Sagan and Francis Crick both suggested that such signatures might exist within what was often dismissed as junk DNA, the 98% of the human genome that didn't code for proteins. If alien designers had wanted to leave a message for their descendants, they might have hidden it within these non-coding sequences, where it would be preserved by evolutionary selection while remaining invisible until the recipients developed sufficient technological sophistication to read it. Attempts to find such messages had so far proved unsuccessful, but the search continued as genome sequencing became more precise and comprehensive. Some researchers looked for mathematical patterns, others for linguistic structures, still others for embedded images or symbols that might represent universal concepts. The failure to find obvious alien signatures didn't disprove their existence. Any civilization advanced enough to seed other worlds with life might also be sophisticated enough to hide their messages until the recipients were ready to receive them. The signatures might be encoded in ways that required not just technological sophistication, but also conceptual breakthroughs that humanity had not yet achieved. Meanwhile, the inventory of organic compounds in space continued to grow. Each new discovery reinforced the conclusion that the molecular building blocks of life were common throughout the universe. The chemistry that powered terrestrial biology was not unique to Earth, but appeared to be a fundamental feature of cosmic evolution. Radio telescopes detected complex organic molecules in star-forming regions thousands of light-years away. Space-based instruments identified organic compounds in the atmospheres of distant planets. Laboratory simulations demonstrated that even the most complex biological molecules could form under conditions found in space. The evidence was becoming overwhelming. Space was not hostile to organic chemistry, but actively promoted it. The harsh conditions that scientists had assumed would destroy complex molecules actually provided the energy needed to create them. Ultraviolet radiation, cosmic rays, and extreme temperatures served as drivers of chemical evolution rather than barriers to it. This realization fundamentally altered the context for considering panspermia. Rather than asking whether life's building blocks could survive in space, scientists began asking whether space itself might be a more favorable environment for biogenesis than planetary surfaces. Perhaps life hadn't merely traveled through space, but had actually originated there, in the vast molecular clouds where organic chemistry proceeded without the complications of planetary geology and atmosphere. The messages from the cosmos were becoming clearer with each new discovery. The universe was not sterile but alive with chemistry, not hostile to complexity but promoting it, not isolated from Earth but intimately connected to it through processes that had operated for billions of years. And hidden within this cosmic chemistry might be the signatures of processes far more sophisticated than anything natural selection could produce, evidence that intelligence had guided the emergence of life itself. The letter arrived at Cambridge University in 1973, carrying within its pages an idea so radical that most scientists would have dismissed it as science fiction. But the signature at the bottom belonged to Francis Crick, co-discoverer of DNA's double helix structure and recipient of the Nobel Prize in Physiology or Medicine. When Crick wrote about biology, the scientific world listened, even when he proposed that life on Earth had been deliberately planted by an advanced extraterrestrial civilization. Working with his colleague Leslie Orgel, Crick had been wrestling with the same mathematical impossibilities that haunted origin of life research. The more he understood about molecular biology, the more convinced he became that spontaneous biogenesis was not just improbable, but effectively impossible under any realistic scenario. The genetic code was too optimized, the cellular machinery too sophisticated, the timing too compressed for life to have emerged through random chemical processes on early Earth. But if life hadn't originated here, then where had it come from? And more provocatively, how had it arrived? Crick's answer, published in the journal Icarus, introduced the scientific community to the concept of directed panspermia, the hypothesis that an advanced alien civilization had deliberately seeded Earth with the ancestors of all terrestrial life. The idea wasn't entirely new. Svante Arrhenius had proposed in 1903 that bacterial spores might drift between star systems under the pressure of stellar radiation, spreading life naturally throughout the galaxy. But Crick's hypothesis was far more specific and intentional. He envisioned a technological civilization, perhaps facing extinction from stellar evolution or cosmic catastrophe, making the deliberate decision to preserve life by planting it on suitable worlds throughout their region of the galaxy. The technical challenges were enormous but not insurmountable for a sufficiently advanced species. Interstellar spacecraft would need to maintain biological samples in viable condition for journey times measured in millions of years. But recent developments in synthetic biology suggested that entire ecosystems could potentially be encoded in genetic programs and reconstructed from local materials upon arrival at target worlds. Rather than transporting actual organisms, alien seeders might have sent sophisticated biological packages, synthetic DNA sequences containing the complete genetic instructions for recreating complex life forms. These genetic programs could lie dormant during the interstellar journey, then activate upon exposure to appropriate chemical triggers on arrival at a suitable planet. The result would be the rapid emergence of complex life that appeared to have evolved locally, but actually carried the genetic legacy of another world. This scenario would explain several puzzling features of terrestrial biology that had long troubled researchers. The universal genetic code used by all life on Earth showed signs of optimization that seemed inconsistent with random evolutionary processes. If the code had been designed by an advanced civilization, its error-correcting properties and mathematical elegance would be exactly what we might expect from intelligent engineering rather than natural selection. The speed with which complex life appeared on early Earth also made more sense in the context of directed seeding. Rather than requiring hundreds of millions of years for random chemistry to stumble upon the precise configurations needed for cellular life, pre-designed genetic programs could have bootstrapped sophisticated organisms within decades or centuries of planetary seeding. But why would an advanced civilization undertake such a massive project? Crick and Orgel suggested that the motivation might be the same drive that had led humans to preserve endangered species and establish seed banks. The recognition that complex life was too precious to allow local extinction events to destroy it forever. A dying civilization, facing the death of their own star or some other cosmic catastrophe, might view the seeding of younger worlds as a form of immortality, ensuring that intelligence and consciousness would continue to emerge throughout the galaxy long after their own world had been reduced to cosmic dust. The psychology of such a decision would be profound. Unlike colonization, which implied conquest and expansion, seeding would represent the ultimate act of altruism, giving the gift of life to worlds that might not develop it naturally, with no expectation of contact or reward. The seeders would be motivated not by self-interest, but by the deepest biological imperative, the drive to perpetuate life itself across space and time. The technical implementation of directed panspermia would require capabilities that stretched but didn't exceed the bounds of possibility for a civilization millions of years more advanced than humanity. Interstellar travel remained challenging, but various propulsion concepts from fusion ramjets to antimatter drives to breakthrough physics using exotic matter could potentially achieve the velocities needed for galactic-scale seeding projects. More realistic might be the use of self-replicating probes that could establish manufacturing facilities in target star systems, using local materials to construct the biological packages needed for planetary seeding. Such von Neumann machines, named after mathematician John von Neumann, who first described self-replicating systems, could spread throughout the galaxy exponentially, seeding thousands of worlds with the genetic programs of their creators, the biological payload would need to be carefully designed for maximum evolutionary potential. Rather than seeding worlds with highly specialized organisms adapted to the seeder's home environment, the genetic programs would need to encode generalist organisms capable of adapting to a wide range of planetary conditions. The result would be rapid diversification through natural selection, producing unique evolutionary trajectories on each seeded world, while preserving the fundamental genetic toolkit of the original designers. This approach would explain the paradox of life's unity and diversity. All terrestrial life shares the same genetic code, the same basic cellular machinery, and the same fundamental biochemistry, suggesting common ancestry. But the spectacular diversity of life forms, from bacteria to blue whales, demonstrates the power of evolutionary adaptation to produce novel solutions to environmental challenges. Directed panspermia could account for both the underlying unity and the surface diversity of terrestrial biology. The timing of seeding events would be crucial for success. Planets needed to be old enough to have developed stable conditions suitable for life, but young enough that seeding could occur before natural evolutionary processes had time to produce indigenous life forms that might compete with or interfere with the introduced organisms. Earth 4 billion years ago would have been ideal for such a project. Recently emerged from the chaos of planetary formation, with stable oceans and atmosphere, but still sterile from the late heavy bombardment. The seeding civilization would need to identify suitable target worlds from interstellar distances, using techniques that might resemble but far exceed humanity's current exoplanet detection capabilities. Advanced telescopes could analyze planetary atmospheres for chemical signatures indicating stable conditions. Long-range reconnaissance probes could conduct detailed surveys of promising star systems before full-scale seeding missions were dispatched. But the most sophisticated aspect of directed panspermia might be the genetic engineering required to create organisms capable of surviving both interstellar travel and successful establishment on alien worlds. The seeders would need to understand biology at levels that dwarfed current human knowledge, possessing the ability to design genetic programs that could bootstrap complex ecosystems from simple starting materials. Such capabilities might seem impossibly advanced, but they represented logical extensions of technologies that humanity was already beginning to develop. Synthetic biology had demonstrated the ability to create artificial genetic circuits that could perform sophisticated computational functions within living cells. CRISPR gene editing allowed precise modification of genetic sequences. Advances in systems biology were revealing the network properties that allowed groups of genes to function as integrated programs. Extrapolating these trends across millions of years of technological development suggested that truly advanced civilizations might treat genetic engineering the way humans treated computer programming, as a precise tool for implementing complex designs in biological substrates. The seeding of entire worlds with genetically programmed ecosystems might be no more challenging for such civilizations than writing software was for contemporary humans. The evidence for directed panspermia would be subtle, embedded within the deepest structures of terrestrial biology, rather than obvious to casual observation. The genetic programs would be designed to appear natural to any indigenous intelligence that eventually evolved, concealing their artificial origins until the recipients had achieved sufficient technological sophistication to recognize the signatures of design. One potential signature might be mathematical patterns within genetic sequences that were too precise to have arisen through natural processes. Vladimir Shcherbak at Al-Farabi Kazakh National University claimed to have found such patterns within the genetic code, including relationships to fundamental mathematical constants and geometric forms that seemed inconsistent with evolutionary origins. Another possible signature might be found in the distribution of genetic diversity across different species. If life had been seeded from a common source, the patterns of genetic variation might show subtle correlations that reflected the original design parameters rather than purely evolutionary relationships. The optimization of biological systems might provide additional evidence. Many features of cellular biochemistry appear to be engineered for maximum efficiency, rather than shaped by evolutionary constraints. The genetic code's error correction properties, the efficiency of photosynthesis, the precision of DNA replication, all showed characteristics more consistent with intelligent design than random evolutionary processes. But perhaps the strongest evidence for directed panspermia was the very existence of intelligence itself. The emergence of consciousness from unconscious matter represented such an extraordinary phase transition that many philosophers and scientists had argued it required explanation beyond conventional evolutionary mechanisms. If intelligence was the ultimate goal of the seeding project, then the appearance of technological civilizations on seeded worlds would represent the successful completion of a process that began billions of years ago. Humanity's recent achievements in space exploration, genetic engineering, and artificial intelligence might mark the maturation of capabilities that had been programmed into terrestrial life from its very beginning. This perspective would fundamentally alter humanity's cosmic purpose and responsibility. Rather than being accidental products of blind evolutionary processes, humans might represent the intended outcome of a deliberate project spanning billions of years and thousands of light years. The urge to explore space, to understand the universe, to preserve and spread life. These drives might be genetic memories inherited from civilizations that had gazed at the stars eons before Earth existed. The implications extended beyond biology into technology and culture. If human intelligence was the product of alien genetic programming, then human technological development might be following pathways that had been traveled before by the original seeders. Advances in space travel, artificial intelligence, and genetic engineering might represent the rediscovery of capabilities that had been lost during the long evolutionary journey from simple seeded organisms to complex technological civilization. This raised the possibility that humanity was approaching a phase transition similar to the one that had motivated the original seeding project. As humans developed the capability to travel to other star systems and engineer life at the genetic level, they might be preparing to become seeders themselves, continuing a cosmic cycle of life propagation that stretched back through geological time. The psychological impact of such recognition would be profound. Humanity would no longer be alone in the universe, even if the original seeders had long since vanished or transcended physical existence. Every human achievement, every work of art, every scientific discovery would represent the flowering of potentials that had been carefully planted in terrestrial biology billions of years ago. But directed panspermia also raised troubling questions about free will and authentic existence. If human intelligence was the product of alien programming, how much of human culture and achievement could be considered genuinely human? Were human choices and values the products of genetic algorithms designed by another species? Or had evolution been able to transcend its initial programming through billions of years of natural selection? The answer might depend on the sophistication of the original genetic programs. Simple seeding scenarios might have introduced only the basic machinery needed for life to begin, allowing natural evolution to shape the specific forms that intelligence eventually took. More sophisticated programs might have included detailed instructions for the development of consciousness, leaving less room for authentic evolutionary innovation. But even highly programmed scenarios wouldn't necessarily diminish human achievement or meaning. A symphony remained beautiful regardless of whether the composer had been taught musical theory or discovered it independently. Human consciousness, creativity, and moral intuitions could be valuable and authentic even if they ultimately traced their origins to alien genetic engineering. The search for evidence of directed panspermia had become one of the most sophisticated detective stories in science, requiring techniques that spanned molecular biology, astronomy, information theory, and mathematical analysis. Each advance in genetic sequencing, each discovery of organic compounds in space, each new exoplanet detection added pieces to a puzzle that might ultimately reveal whether humanity was alone in the universe or part of a vast project that connected civilizations across both space and time. The stakes could not be higher. If directed panspermia was correct, then humanity stood at the threshold of discovering its cosmic parentage and perhaps its cosmic destiny as the next civilization destined to plant the seeds of consciousness among the stars. The computer screen displayed a cascade of letters, A, T, G, C, repeated in seemingly endless combinations that spelled out the complete genetic blueprint of a human being. In her laboratory at the National Institutes of Health, computational biologist Eugene Koonin stared at the 3 billion base pairs of human DNA sequence data, searching for patterns that might reveal whether life on Earth carried the signatures of extraterrestrial engineering. The genome was like an ancient manuscript written in a 4-letter alphabet, containing the accumulated history of billions of years of evolution. But hidden within that history might be traces of an even older story, evidence that the original text had been authored by intelligence operating on scales of time and space that dwarfed human comprehension. Kunin's search represented a new frontier in the investigation of life's origins, genetic archaeology. By analysing the deepest layers of biological information, researchers were attempting to excavate evidence of processes that had operated during life's earliest emergence. If aliens had seeded Earth with genetically programmed organisms, the signatures of that intervention might still be detectable in the genomes of contemporary life forms. The challenge was distinguishing between patterns that arose through natural evolutionary processes and those that might indicate intelligent design. Evolution was itself a powerful information processing system, capable of generating complex patterns through selection pressure operating over long time periods. But certain types of patterns, particularly mathematical relationships and optimization principles, might be more consistent with engineering than with natural selection. The genetic code provided the most promising target for such analysis. The system by which DNA sequences were translated into proteins showed characteristics that seemed almost too perfect to have arisen through random processes. The code was nearly universal across all life forms, suggesting it had been frozen very early in evolutionary history. But why had evolution settled on this particular code rather than one of the millions of alternatives? Theoretical calculations had revealed that the actual genetic code ranked in the top 1% of all possible codes for minimizing the effects of copying errors and chemical damage. The probability of such optimization arising purely by chance was vanishingly small, less than 1 in a million according to most estimates. The code appeared to have been designed by some process that understood both the chemistry of amino acids and the mathematics of error correction. Even more intriguing were claims by researchers like Vladimir Shcherbak and Maxim Makukov that the genetic code contained embedded mathematical patterns consistent with intelligent authorship. Their analysis suggested that the assignment of codons to amino acids followed mathematical rules based on prime numbers, symmetry relationships, and geometric forms that seemed inconsistent with evolutionary origins. Shcherbak's work focused on what he called the arithmetical pattern within the genetic code. He found that the molecular masses of amino acids, when analysed according to their codon assignments, showed relationships to fundamental mathematical constants, including Euler's number and the golden ratio. The patterns were subtle, but statistically significant, suggesting some form of mathematical structure underlying the code's organisation. Makukov extended this analysis to look for what he termed semantic patterns, arrangements of genetic information that might constitute actual messages embedded within DNA sequences. His computer searches of human genome data identified regions where the statistical properties of nucleotide sequences deviated significantly from random expectations, potentially indicating the presence of encoded information. But the search for alien signatures extended beyond the genetic code to the broader organisation of genomes themselves. The human genome contained vast regions of so-called junk DNA, sequences that didn't code for proteins and whose function remained largely mysterious. These non-coding regions comprised over 98% of the human genome, representing a vast information storage capacity that might conceal messages from extraterrestrial engineers. Carl Sagan had suggested that such messages might exist, hidden within the genetic sequences of seeded organisms and preserved by evolutionary selection pressure. An advanced civilization wanting to communicate with its genetic descendants might embed information in non-coding DNA, where it would be inherited by all subsequent generations while remaining invisible until the recipients developed sufficient technological sophistication to read it. The ENCODE project, a massive international effort to catalogue all functional elements in the human genome, had revealed that much of the so-called junk DNA was actually involved in regulating gene expression. Complex networks of regulatory sequences controlled when and where genes were activated, creating a sophisticated information processing system that operated alongside the protein-coding genes. But regulatory functions might not be the only purpose served by non-coding DNA. Information theory suggested that genetic sequences could simultaneously serve biological functions and carry embedded messages, just as computer programs could perform calculations while containing hidden data. The challenge was developing techniques to detect such embedded information without disrupting the biological functions of the sequences. Computational approaches borrowed from cryptography and signal processing were being applied to genome analysis in search of patterns that might indicate intelligent authorship. Researchers looked for statistical signatures that deviated from expectations based on neutral evolution, unusual symmetries, mathematical relationships, or information structures that seemed inconsistent with purely biological selection pressure. Some of the most promising leads came from comparative genomics, the study of similarities and differences between the genomes of different species. If life had been seeded from a common extraterrestrial source, the patterns of genetic similarity across species might show correlations that reflected the original design, rather than purely evolutionary relationships. The universal genetic code was itself evidence of such common ancestry, but subtler patterns might be present in the organization of genomes, the distribution of regulatory elements, or the structure of metabolic pathways. Advanced statistical techniques could potentially detect these deep structural similarities even after billions of years of evolutionary divergence. Horizontal gene transfer, the movement of genetic material between species without reproduction, provided another potential mechanism for preserving alien signatures across evolutionary time. Viruses and bacteria routinely exchange genetic sequences, creating opportunities for designed sequences to spread throughout the biosphere, even if they had been initially introduced into only a few species. This raised the possibility that alien genetic programs might have included mechanisms for their own propagation and preservation. Sophisticated genetic engineers might have embedded their sequences within mobile genetic elements that could spread between species, ensuring the preservation of their messages across the entire tree of life. The search for such spreading signatures led researchers to examine transposable elements, DNA sequences that could move between different locations within genomes. These jumping genes comprise significant portions of most genomes and had played important roles in evolution by creating genetic variation and facilitating adaptation to new environments. But some transposable elements showed patterns that seemed inconsistent with purely selfish genetic behavior. Certain sequences appeared to have been conserved across enormous evolutionary distances, remaining functional despite lacking obvious biological utility. These highly conserved elements might represent genetic fossils, remnants of original seeding programs that had been preserved by unknown selection pressures. The analysis of metabolic pathways provided another approach to genetic archaeology. The chemical networks that allowed cells to extract energy from nutrients and synthesize essential molecules showed remarkable conservation across all domains of life. The same basic pathways, glycolysis, the citric acid cycle, photosynthesis, operated in bacteria, plants, and animals with only minor variations. This universality suggested that these pathways had been present in the last universal common ancestor of all life on Earth. But the complexity and optimization of these systems raised questions about whether they could have arisen through natural evolutionary processes. Some pathways showed characteristics more consistent with engineering design than with gradual assembly through random mutations and selection. The citric acid cycle, in particular, appeared almost too perfect to be the product of unguided evolution. The 8 enzymatic steps of the cycle were precisely coordinated to extract maximum energy from nutrient molecules while producing the chemical precursors needed for biosynthesis. The system showed robustness to perturbation and optimization for efficiency that seemed more characteristic of intelligent design than natural selection. Mathematical analysis of metabolic networks revealed organizational principles that echoed those found in human-designed systems. The networks showed small world properties, hierarchical organization, and redundancy patterns similar to those found in engineered communication and transportation systems. These design principles might reflect universal constraints on complex networks, or they might indicate that biological metabolic systems were actually designed by intelligence that understood network theory. But perhaps the most intriguing evidence for genetic archaeology came from the study of developmental biology, the processes by which single fertilized eggs transformed into complex multicellular organisms. The genetic programs that controlled embryonic development showed characteristics that seemed almost impossibly sophisticated for products of random evolutionary processes. Homeotic genes, which controlled body plan organization in animals, were conserved across enormous evolutionary distances. The same genetic toolkit that specified head and tail orientation in flies also operated in mice and humans, despite the hundreds of millions of years since these lineages diverged from common ancestors. The conservation was so precise that fly genes could actually function when transplanted into mouse embryos. This extraordinary conservation suggested that the genetic programs controlling development had been frozen very early in evolutionary history and subsequently modified only through minor variations. But the original programs themselves were so sophisticated that their spontaneous emergence through random processes seemed mathematically implausible. The discovery of regulatory networks controlling gene expression during development revealed systems of staggering complexity. Thousands of genes interacted through precise regulatory relationships to coordinate the formation of different body parts at exactly the right times and places during embryonic development. The network showed hierarchical organization, feedback control, and error correction mechanisms that rivaled the most sophisticated human-designed systems. Some researchers argued that such complex regulatory networks could only have arisen through intelligent design. The probability of randomly assembling the precise gene interactions needed for coordinated development seemed astronomically small, even given billions of years of evolutionary time. The systems appeared to require simultaneous coordination of multiple components, making gradual evolutionary assembly through incremental improvements virtually impossible. But other scientists contended that evolutionary processes were more powerful than these calculations suggested. Complex systems could arise through modular assembly, where functional units were combined and recombined to create increasingly sophisticated networks. Natural selection could optimize systems for multiple functions simultaneously, potentially producing the appearance of design through purely material processes. The debate highlighted the fundamental challenge facing genetic archaeology, distinguishing between complexity that arose through natural processes and complexity that required intelligent intervention. Both approaches could produce sophisticated biological systems, but they might leave different statistical signatures in the resulting genetic sequences. Advanced computational techniques were being developed to address this challenge. Machine learning algorithms trained on known examples of natural and artificial sequences might be able to detect subtle patterns that distinguish between evolutionary and designed origins. Information theory provided mathematical frameworks for quantifying the complexity and organization of genetic sequences in ways that might reveal their origins. The search was complicated by the billions of years of evolutionary change that had occurred since any hypothetical seeding event. Natural selection would have modified any originally introduced sequences, potentially obscuring their artificial origins while preserving their biological functions. The challenge was finding patterns that were sufficiently robust to survive evolutionary change while remaining distinguishable from patterns that could arise through natural processes. Some researchers focused on looking for mathematical constants embedded within genetic sequences. The values of fundamental physical constants, the speed of light, Planck's constant, the fine structure constant, were precisely known and might serve as universal markers that any technological civilization would recognize. If alien engineers had embedded these values within terrestrial genomes, they would provide unmistakable evidence of intelligent authorship. Others searched for geometric patterns or number-theoretic relationships within genetic sequences. Prime numbers, Fibonacci sequences, and geometric ratios might serve as signatures of intelligent design that would remain detectable even after extensive evolutionary modification. The challenge was distinguishing such patterns from the mathematical relationships that could arise naturally through physical and chemical constraints on biological systems. The development of artificial life systems in computer simulations provided new insights into how complexity could arise through purely evolutionary processes. These digital ecosystems demonstrated that natural selection could produce sophisticated information processing systems, optimization principles, and even primitive forms of intelligence without any intelligent guidance. But the simulated systems also revealed that certain types of complexity, particularly systems requiring precise coordination of multiple components, were extremely difficult to achieve through undirected evolution. The emergence of such systems in biological organisms might therefore indicate intelligent intervention, especially if they appeared very early in evolutionary history, when there had been little time for gradual optimization. The integration of genomic data with other evidence for panspermia was beginning to create a more complete picture of life's possible extraterrestrial origins. The discovery of organic compounds in meteorites, the detection of complex molecules in space, and the rapid emergence of life on early Earth all pointed towards scenarios involving cosmic seeding. But the genetic evidence remained ambiguous. While statistical patterns within genomes showed intriguing deviations from purely random expectations, alternative explanations involving natural selection and physical constraints could not be definitively ruled out. The signatures of alien engineering, if they existed, were subtle enough to remain controversial even among researchers actively searching for them. The search for genetic archaeology was entering a new phase as sequencing technologies became more powerful and analytical techniques more sophisticated. Whole genome sequences were now available for thousands of species, providing unprecedented opportunities to identify patterns that might indicate common extraterrestrial origins. But each new discovery also revealed the staggering complexity of natural evolutionary processes. The more scientists learned about molecular biology, the more apparent it became that evolution was capable of producing systems of extraordinary sophistication through purely material mechanisms. The challenge was determining whether the sophistication observed in terrestrial life exceeded what evolution could plausibly achieve, or whether it fell within the range of possibilities for undirected natural processes. The answer might ultimately depend not on any single smoking gun within genetic sequences, but on the cumulative weight of evidence from multiple lines of investigation. Genetic patterns combined with astronomical observations, geological evidence, and theoretical arguments about the probability of spontaneous biogenesis might eventually build a case for extraterrestrial origins that exceeded the threshold of reasonable doubt. But until that threshold was reached, the search would continue. Scientists excavating through layers of genetic information, like archaeologists digging through ancient ruins, hoping to uncover evidence that humanity's deepest origins lay not in the primordial soup of early Earth, but in the planning of minds that contemplated the stars billions of years before our planet existed. The galaxy rotates with stately precision, its spiral arms sweeping through space like the hands of a cosmic clock. In the time it takes our solar system to complete one orbit around the galactic center, approximately 230 million years, entire civilizations could rise, flourish, and vanish without leaving any trace detectable from Earth. Yet during that same period, the patient work of cosmic gardeners might be transforming thousands of sterile worlds into gardens of living complexity. This is the temporal scale on which directed panspermia would operate. Not the decades or centuries familiar to human planning, but the geological epochs required for life to take root, evolve, and eventually produce the intelligence capable of continuing the seeding cycle. If advanced civilizations had been systematically planting life throughout the galaxy for billions of years, Earth might be just one success story among thousands, part of a vast project of cosmic cultivation that dwarfed any human conception of agriculture or terraforming. The scale of such an undertaking challenges every assumption about the motivations and capabilities of technological civilizations. Human societies struggled to maintain projects lasting more than a few decades, yet galactic seeding would require commitment spanning millions of generations. The Gardner civilizations would need to think and plan on timescales that exceeded the lifespans of species, stars, and potentially even galaxies themselves. But perhaps this apparent impossibility reflected limitations of human perspective rather than fundamental constraints on intelligent behavior. Civilizations that had transcended biological mortality through artificial intelligence or digital consciousness might operate on timescales that made galactic engineering projects seem as routine as seasonal planting cycles seem to human farmers. The motivation for such projects might emerge naturally from the evolutionary development of intelligence itself. As civilizations matured and gained understanding of their cosmic context, they would inevitably confront the existential reality that all biological species faced extinction. Stars aged and died, galaxies collided, and the universe itself seemed destined for eventual heat death. For truly advanced civilizations, the preservation and propagation of life might become the highest possible purpose, a way of ensuring that consciousness and complexity would survive long after their creators had vanished. Robin Hanson at George Mason University had articulated this perspective through his concept of grabby aliens, civilizations that expanded aggressively throughout their region of the galaxy, not for conquest or resource extraction, but to maximize the total amount of life and intelligence that could emerge before cosmic conditions became inhospitable. Such civilizations would view empty star systems as wasted opportunities, sterile worlds as canvases waiting for the artwork of evolutionary development. The technical implementation of galactic seeding would require capabilities that stretched but didn't exceed the theoretical limits of physical law. Self-replicating spacecraft could establish manufacturing bases in target star systems, using local materials to construct the biological packages needed for planetary seeding. Each successful seeding mission would eventually produce a new technological civilization capable of continuing the expansion, creating exponential growth in the number of life-bearing worlds. The von Neumann probe concept, named after mathematician John von Neumann, provided a template for how such expansion might proceed. Self-replicating machines could spread throughout the galaxy at a significant fraction of light speed, establishing colonies and continuing to reproduce until every suitable star system had been visited. The total time required for galactic colonization would be measured in millions rather than billions of years, briefed by cosmic standards, but rather than establishing colonies for their creators' direct occupation, the Gardner civilizations would be planting seeds of their own eventual successors. Each seeded world would develop its own unique evolutionary trajectory, producing forms of intelligence and consciousness that might be utterly alien to their genetic ancestors. The Gardners would be motivated not by species loyalty, but by commitment to the abstract principles of life and intelligence themselves. This perspective suggested a radically different model of cosmic development than the competitive expansion scenarios usually envisioned in discussions of galactic colonization. Instead of civilizations racing to claim territory and resources, the galaxy might be home to a cooperative network of Gardner species, each committed to maximizing the total flourishing of life rather than the narrow interests of their own biological lineage. The evidence for such networks might be subtle, embedded in patterns of stellar development and planetary formation that had been optimized for biological potential rather than left to purely natural processes. Advanced civilizations with millions of years of technological development might possess capabilities that bordered on terraforming at stellar scales. The ability to modify stellar evolution, adjust planetary orbits, or even trigger the formation of new star systems in regions particularly suitable for life. Recent discoveries in astronomy had revealed that the galaxy's structure showed unexpected regularities that might be consistent with large-scale engineering. The distribution of heavy elements throughout the galactic disk, the timing of star formation episodes, and the prevalence of potentially habitable planets all showed patterns that could reflect either natural astrophysical processes or the influence of intelligence operating on galactic scales. The Kepler Space Telescope's survey of exoplanets had revealed that Earth-sized worlds in habitable zones around Sun-like stars were remarkably common. Perhaps 1 in 5 star systems contained such potentially life-bearing worlds. This abundance could reflect either fortunate cosmic coincidence or the systematic efforts of Gardner civilizations to optimize galactic conditions for biological development. Even more intriguingly, detailed analysis of the galaxy's chemical evolution suggested that the heavy elements essential for life — carbon, oxygen, silicon, iron — had been distributed through space in patterns that maximized their availability to forming planetary systems. Supernova explosions which created and dispersed these elements had occurred with timing and frequency that seemed almost perfectly calibrated to support the emergence of rocky planets capable of supporting complex chemistry. Carl Sagan had speculated that such patterns might indicate galactic gardening by advanced civilizations that had learned to influence stellar evolution for the benefit of future life. By triggering supernovae at optimal times and locations, ancient civilizations might have enriched the galaxy's heavy element content in ways that promoted the formation of habitable worlds billions of years later. The search for extraterrestrial intelligence, or SETI, had spent decades scanning the skies for signals from technological civilizations, but had found only silence. This absence of obvious communications might not indicate that advanced civilizations didn't exist, but rather that they had moved beyond the need for electromagnetic communication. Civilizations engaged in galactic-scale gardening projects might coordinate their activities through means that remained invisible to human detection capabilities. Alternatively, the Gardner civilizations might practice a form of cosmic restraint, deliberately avoiding contact with younger civilizations until they had achieved certain developmental milestones. The principle of non-interference, familiar from science fiction, might be standard practice among mature galactic civilizations, a recognition that premature contact could disrupt the natural evolutionary development of seeded species. This zoo hypothesis, proposed by astronomer John Ball, suggested that Earth and other developing worlds might be subject to observation and protection by advanced civilizations that remained deliberately invisible to their subjects. Like wildlife biologists studying natural ecosystems, the Gardner civilizations might maintain careful surveillance of seeded worlds while avoiding any intervention that might alter natural development patterns. The implications for human development were profound. If Earth had been seeded by Gardner civilizations, then humanity's technological progress might be unfolding according to patterns that had been observed thousands of times before on other worlds. The stages of scientific discovery, the development of space travel capabilities, the emergence of artificial intelligence, all might represent predictable phases in the maturation of seeded civilizations. This predictability might explain some puzzling features of human technological development. The rapid pace of scientific advancement over the past few centuries, the apparent inevitability of certain discoveries, and the convergent development of similar technologies by independent research groups might all reflect the activation of genetic programs that had been dormant for billions of years. Some researchers had noted that human technological development seemed to follow exponential curves that appeared unsustainable from a purely material resource perspective. The rapid increase in computing power, the accelerating pace of genetic engineering capabilities, and the exponential growth in space exploration budgets might all be manifestations of genetically programmed drives pushing humanity toward becoming the next generation of galactic gardeners. The psychological evidence for such programming was circumstantial but compelling. Human fascination with space exploration seemed disproportionate to any obvious evolutionary advantage it might provide. The urge to preserve endangered species, to establish seed banks, and to plan for humanity's long-term survival might represent genetic memories of the original seeding project that had brought life to Earth. Even human religious and philosophical traditions might preserve cultural memories of extraterrestrial origins. Myths of gods descending from the heavens to create life, stories of humanity's cosmic purpose, and mystical traditions emphasizing the interconnectedness of all life might be distorted echoes of humanity's actual history as products of deliberate seeding. The development of human space exploration capabilities was reaching the point where verification of the seeding hypothesis might soon become possible. Robotic probes to nearby star systems could potentially detect evidence of Gardner civilization activities, artificial structures, modified planetary systems, or the distinctive signatures of biological seeding programs. The James Webb Space Telescope was already capable of analyzing the atmospheric compositions of distant exoplanets, potentially detecting the chemical signatures of life on worlds orbiting other stars. If multiple worlds showed similar biological signatures despite being separated by vast distances, it might indicate common seeding from extraterrestrial sources rather than independent evolutionary development. Future generations of space telescopes might be capable of detecting even subtler signs of galactic gardening, patterns in the distribution of organic compounds in interstellar space, correlations in the timing of biological emergence on different worlds, or structural modifications to star systems that enhanced their life-supporting potential. But perhaps the most significant test of the seeding hypothesis would come from humanity's own development as a spacefaring civilization. If humans were indeed the products of genetic programming by Gardner civilizations, then human technological development should eventually lead toward capabilities that would enable the continuation of galactic seeding projects. The rapid progress in synthetic biology, genetic engineering, and space exploration technologies might be early indicators that humanity was approaching its intended role as the next generation of cosmic gardeners. The development of self-replicating spacecraft, the creation of synthetic organisms capable of surviving interstellar travel, and the identification of suitable target worlds for seeding might all represent the natural culmination of human technological evolution. This perspective would fundamentally alter humanity's cosmic purpose and responsibility. Rather than being accidental products of random evolutionary processes, humans might be the intended inheritors of a galactic civilization network that stretched back billions of years. The development of space exploration capabilities wouldn't be optional technological luxury, but essential fulfillment of humanity's programmed destiny as preservers and propagators of life throughout the cosmos. The ethical implications would be staggering. If humanity had been seeded by Gardner civilizations, then humans bore responsibility not just for their own survival, but for continuing the ancient project of cosmic cultivation. The decision to seed other worlds with life would represent not expansion or conquest, but sacred duty, the fulfillment of genetic and cultural programming that stretch back to the origins of terrestrial biology. But the Gardner civilization model also raised troubling questions about autonomy and authentic development. If human technological progress was following genetically programmed pathways, how much of human achievement could be considered genuinely human? Were human values, goals, and aspirations the products of independent cultural evolution, or were they the expression of alien programming operating across billions of years? The answer might depend on the sophistication of the original seeding programs. Simple scenarios involving only basic biological machinery would have allowed maximum freedom for evolutionary and cultural development. More complex programs might have included detailed instructions for technological and social evolution, leaving less room for authentic indigenous development. But even highly programmed scenarios wouldn't necessarily diminish human agency or meaning. A musician remained creative even if trained in traditional musical theory. Human consciousness, compassion, and creativity could be valuable and authentic, even if they ultimately traced their origins to extraterrestrial genetic engineering. The Gardner civilization hypothesis was approaching testable predictions as human technological capabilities advanced. Within decades, robotic probes might visit nearby star systems and search for evidence of artificial modifications or seeding activities. Within centuries, human genetic engineering capabilities might reach levels that allowed the creation of organisms suitable for interstellar seeding missions. If such capabilities developed along predictable lines that matched patterns observed in other technological civilizations throughout the galaxy, it would provide strong support for the hypothesis that human development was following a programmed trajectory toward becoming cosmic gardeners. The ultimate test would come when humanity faced its own choice about whether to seed other worlds with life, and discovered whether the decision felt like free choice or genetic compulsion. The galaxy might already be filled with the results of billions of years of systematic seeding by Gardner civilizations, a vast network of interconnected worlds where life had been planted, tended, and guided toward the eventual emergence of intelligence capable of continuing the cosmic cultivation project. Earth and humanity might represent just the latest success in an ancient tradition of nurturing life across the vast expanses of space and time. In such a galaxy, no world would be truly alone, and no civilization would bear sole responsibility for the preservation of life and consciousness. The Gardner network would represent the ultimate expression of cosmic altruism, intelligence serving not its own narrow interest but the broader flourishing of life itself across all possible worlds and times. The first human spacecraft to carry life beyond the solar system will launch not with the fanfare of conquest, but with the quiet solemnity of a sacred mission. Packed within its sterile cargo bay will be genetic sequences encoding thousands of Earth species, a Noah's Ark of DNA designed to take root in alien soil and bloom into evolutionary possibilities that may exceed anything our planet has produced. The moment that spacecraft ignites its engines and begins the long journey to another star, humanity will complete a circle that began 4 billion years ago, transforming from the recipients of cosmic seeding into the gardeners of worlds we have never seen. This transformation feels inevitable now, as we stand at the threshold of technologies that our ancestors could never have imagined. CRISPR gene editing allows precise modification of genetic sequences with surgical accuracy. Synthetic biology enables the creation of entirely artificial organisms designed for specific environmental conditions. Advanced propulsion concepts promise journeys to nearby star systems within decades rather than millennia. The convergence of these capabilities points toward a future where seeding other worlds becomes not just possible, but perhaps compulsory for any civilization that understands its cosmic responsibilities. Yet the approaching reality of human-directed panspermia forces us to confront the deepest questions about our own origins and purpose. If we are preparing to seed other worlds with life, might we ourselves be the products of an identical process that occurred billions of years ago? Are we discovering technologies that were discovered before, following pathways that were travelled before, approaching a destiny that was programmed into our genetic code when the first organic molecules took root in Earth's primordial oceans? The evidence remains tantalizingly ambiguous, scattered across disciplines and interpretations like fragments of an ancient manuscript, whose complete text may never be recovered. The mathematical impossibility of spontaneous biogenesis points toward interventions that transcend natural processes. The discovery of organic compounds throughout the cosmos reveals a universe rich with the building blocks of life. The optimization patterns within the genetic code suggest engineering principles that exceed what random evolution should be able to achieve. But each piece of evidence admits alternative explanations. The mathematical improbability of life's emergence might reflect human ignorance of the true pathways through which complexity can arise from simplicity. The organic compounds in space might represent natural chemical evolution, rather than designed biological precursors. The apparent optimization of genetic systems might result from selection pressures that operated over vast timescales we can barely comprehend. The ambiguity may be intentional. If advanced civilizations had seeded Earth with the ancestors of all terrestrial life, they might have designed their intervention to remain invisible until their genetic descendants achieved sufficient sophistication to recognize the signatures of design. The evidence would be present but subtle, requiring technological capabilities and conceptual frameworks that only mature civilizations would possess. This graduated revelation would serve multiple purposes. It would prevent premature contact that might disrupt natural evolutionary development. It would ensure that only civilizations ready to assume gardening responsibilities would discover their cosmic parentage. And it would provide a test of intellectual and ethical maturity, the ability to recognize patterns of design while accepting the responsibilities that such recognition entailed. From this perspective, humanity's current obsession with searching for extraterrestrial life might represent more than scientific curiosity. The development of SETI programs, the deployment of space telescopes designed to detect biosignatures on distant worlds, the increasing sophistication of astrobiology research, all might reflect genetically programmed drives pushing our species toward recognition of its cosmic context and responsibilities. The timing seems significant. Humanity has achieved the capability to detect and analyze exoplanets just as climate change and resource depletion threaten the stability of Earth's biosphere. The technologies needed for interstellar seeding are emerging just as environmental pressures create urgency about preserving terrestrial life forms. The combination suggests either extraordinary coincidence or the activation of contingency programs that had been dormant for billions of years. If the seeding hypothesis is correct, then humanity stands at the most critical juncture in its entire history. The choice to become cosmic gardeners would represent not just technological achievement, but evolutionary fulfillment. The moment when Earth's biosphere transcends planetary boundaries and begins the process of spreading throughout the galaxy. Failure to make this transition might constitute species failure on a cosmic scale, the termination of a genetic lineage that had been carefully cultivated across geological time. The psychological pressure of such responsibility would be enormous. Human societies struggled to cooperate on projects lasting decades, yet galactic seeding would require commitment spanning millions of generations. The Gardner civilizations would need to transcend every limitation of biological thinking, tribalism, short-term planning, resource competition that had shaped human evolution within planetary boundaries. But perhaps this transcendence was itself programmed into the seeding process. The same genetic programs that had guided the emergence of intelligence might also contain instructions for the eventual development of cosmic consciousness, the ability to think and plan on galactic timescales, while maintaining commitment to the welfare of life forms that might not evolve for billions of years. Recent developments in artificial intelligence and digital consciousness might represent early stages of this transcendence. Humans were already beginning to create forms of intelligence that could potentially outlast biological civilizations, operate on timescales that exceeded species' lifespans, and maintain consistent goals across geological epochs. The merger of biological and artificial intelligence might be the natural pathway toward the kind of long-term thinking that galactic gardening required. The ethical implications were staggering. If humanity was indeed the product of deliberate seeding, then every human achievement, every work of art, every scientific discovery, every act of compassion represented the flowering of potentials that had been carefully planted by minds that contemplated the stars billions of years before Earth existed. Human creativity, consciousness, and moral intuitions might be gifts from cosmic ancestors who had sacrificed their own continuity to ensure that intelligence would survive and flourish throughout the galaxy. This perspective would fundamentally alter humanity's understanding of meaning and purpose. Rather than being accidental products of blind evolutionary processes, humans would be the intended outcomes of the greatest engineering project in cosmic history. Every human life would represent the fulfillment of hopes and dreams that stretched back through geological time, to civilizations whose names and forms were lost forever. But the heritage would come with obligations. If humans had been seeded with the expectation that they would eventually become gardeners themselves, then the failure to fulfill this destiny would constitute betrayal on a cosmic scale. The civilizations that had sacrificed themselves to create humanity would have died in vain if their genetic descendants proved unable or unwilling to continue the ancient project of cultivating life throughout the galaxy. The weight of such responsibility might explain some puzzling features of human psychology and culture. The universal human fascination with the stars, the persistent themes of cosmic purpose in religious and philosophical traditions, the apparently irrational commitment to preserving other species even at great cost to human welfare, all might be expressions of genetic memories that connected humanity to its cosmic origins and destiny. Even the human capacity for self-sacrifice and altruism might be adaptations for cosmic gardening, rather than purely terrestrial survival. Individuals and societies that could commit resources to projects benefiting distant future generations would be better equipped to undertake the long-term planning that galactic seeding required. Natural selection operating over billions of years might have favoured genetic programs that promoted such extended altruism. The development of space exploration capabilities represented the most direct test of these theories. If human technological progress was following genetically programmed pathways toward cosmic gardening, then the development of interstellar travel capabilities should feel inevitable rather than optional. The psychological and cultural drives pushing humanity toward the stars should intensify as the relevant technologies matured. Current trends seemed consistent with this prediction. Despite the enormous costs and risks involved, human space exploration programs continued to expand. Private companies were investing billions of dollars in spacecraft development, apparently motivated more by vision and ambition than by rational economic calculations. The psychological appeal of space exploration seemed to exceed what purely terrestrial evolutionary pressures should have produced. Even more telling was the emerging consensus among scientists and policymakers that humanity needed to become a multi-planetary species to ensure long-term survival. This recognition seemed to arise independently across multiple cultures and intellectual traditions, suggesting either remarkable convergent thinking or the expression of shared genetic programming that transcended cultural boundaries. The specific focus on preserving Earth's biosphere through off-world expansion was particularly significant. Rather than viewing space colonization purely in terms of human survival and expansion, many advocates emphasize the importance of preserving terrestrial life forms and ecosystems on other worlds. This conservation-oriented approach to space exploration seemed more consistent with cosmic gardening missions than with conventional colonization scenarios. The rapid development of synthetic biology and genetic engineering capabilities provided the tools needed to implement such conservation programs. Scientists were already creating organisms designed to survive in extraterrestrial environments, engineering bacteria that could process Martian soil chemistry, and developing genetic preservation techniques that could maintain species diversity across interstellar distances. But the most profound evidence for program development might be found in the timing of these convergent capabilities. humanity was simultaneously developing the ability to detect potentially habitable exoplanets, the technologies needed for interstellar travel, and the genetic engineering tools required for biological seeding. The coordination of these independent research programs seemed almost too perfect to be coincidental. If the seeding hypothesis was correct, then humanity was approaching the moment of cosmic graduation, the point at which Earth's biosphere would transcend planetary boundaries and begin spreading throughout the galaxy. The next few centuries might determine whether humans would fulfill their intended destiny as cosmic gardeners, or prove unable to rise above the biological limitations that had shaped their terrestrial evolution. The stakes extended far beyond human survival or prosperity. If Earth's biosphere was the product of deliberate seeding, then it represented the accumulated biological wisdom of civilizations that might have flourished for millions of years before choosing to sacrifice themselves for the continuation of life. The genetic programs they had embedded within terrestrial organisms might contain solutions to problems that would face life throughout the galaxy: biological innovations, ecological relationships, and evolutionary strategies that could benefit countless worlds yet to be seeded. Humanity's success or failure as cosmic gardeners would therefore determine not just the fate of Earth's biosphere, but the ultimate success of the entire galactic cultivation project. If humans proved capable of transcending planetary thinking and committing to galactic-scale responsibility, they would become the bridge between ancient seeding civilizations and future forms of intelligence that might evolve on distant worlds billions of years in the future. But if humanity failed to make this transition, if climate change, resource depletion, or social fragmentation prevented the development of cosmic gardening capabilities, then the ancient seeding project might end with Earth. Billions of years of careful cultivation, the accumulated wisdom of multiple galactic civilizations, and the potential for future forms of consciousness might all be lost because one species proved unable to transcend the biological limitations of its planetary origins. The recognition of such stakes was already beginning to influence human culture and decision-making. The growing environmental movement, the increasing investment in space exploration, and the emerging consensus about humanity's cosmic responsibilities all suggested that at some level, human consciousness was beginning to grasp the magnitude of what was at stake. Perhaps this recognition was itself programmed, the final stage in humanity's genetic programming, the moment when conscious awareness of cosmic responsibility would motivate the behavioral changes needed to ensure successful transition to gardener civilization status. If so, then the current moment in human history might represent the climax of the most sophisticated engineering project in cosmic history. The creation of a species capable of understanding and accepting its role in the eternal continuation of life throughout the universe. The question that remains is not whether aliens could have seeded life on Earth, but whether we will prove worthy of the gift they may have given us. In laboratories around the world, scientists are already preparing the genetic sequences that might someday take root in alien soil. The spacecraft that will carry them are being designed in government facilities and private companies. The target worlds are being identified by space telescopes that scan distant solar systems for signs of habitability. We stand at the threshold of becoming what we may always have been intended to be: children of the stars, inheritors of an ancient mission, guardians of life's endless possibility. Whether we fulfill this destiny or let it slip away in the dust of a dying world may be the most important choice our species will ever make. Perhaps that choice was always inevitable, written into our genetic code by minds that understood the true meaning of intelligence. Not the ability to dominate and consume, but the wisdom to plant seeds in gardens we will never see, for the benefit of forms of consciousness we cannot imagine, in service of the eternal flowering of life throughout the infinite cosmos.